while doing the serum urea estimation, in the kit there will be literature. Okay, I have printed it out, and there is literature, and everything will be written. During exam you will be having doubt. Okay, during practicing and examination, that how we are doing, and the procedure, all that. So it's better you go through the kit literature and know by yourself what we are doing, the importance, procedure, principles. Check it yourself. Okay, for urea estimation we will be doing modified Berthelot method. Do I have to write or modified Berthelot method? No. Okay, urea. Yes, sir. Thank you, sir. This is the structure of urea: CO, NH2 and NH2. Okay, this C comes from carbon dioxide, one N comes from ammonia, another N comes from amide group of aspartate. You have to remember this. If they ask like this, in this structure, which atom is coming from where? So this is from carbon dioxide, this is from ammonia, and another N from amide group of aspartate. Okay, aspartate also is there. In urea cycle it will be in liver, and some of the steps in mitochondria and some of the steps are in cytoplasm of liver cell. Okay, and if you remember the mnemonic, you can remember this one or this one, whatever, but I think this one will be better. Okay, ornithine, citrulline, arginosuccinate and arginine. And ornithine will be in the mitochondria, the step inside the mitochondria, and these three steps will be in the cytoplasm. Okay, and there ATP also is needed. What? This is ornithine, citrulline, arginosuccinate, and this is arginine. The last step will be arginase. From there arginine will be broken down and will form the urea. And steps of urea formation, if they ask, there will be 4 steps. 1, 2, 3, 4. 4 steps in that urea will be formed. First is transamination. Oxidative deamination. Ammonia transport. And last is urea cycle. So how urea is formed? You have to remember that 4 steps: transamination, oxidative deamination, then ammonia will be transported, and last is urea cycle in the liver. Okay, now I can erase it off. Alpha amino acid, it will give rise to alpha keto acid. So transamination of alpha amino acid will form alpha keto acid, and alpha ketoglutarate will work and will form the glutamate. Okay, this is the transamination process. From here, it will be oxidative deamination. Ammonia will be formed and carbon dioxide will join from outside and it will form the urea. Now, can I move on or do you want to write? Urea is forming in liver and excreted through kidney. So, any problem in liver and kidney will affect the, I mean not all, but it can affect the urea. Okay, urea excretion and production also. So, what happens in liver disease, you will see less urea, and in pregnancy also there will be less urea. Pregnancy, I have to find the cause, but one of the cause can be hemodilution. If it's too much water is there, urea content is becoming low. In pregnancy also, urea less. Pregnancy and liver disease and children also, urea can be low. Female also is urea content is lower than the male. Male is more protein catabolism. From protein it is coming amino acid, and from amino acid the urea is forming. So for male also urea is more, and where urea will be more for male, and anywhere where you are taking more protein, you are eating more protein and hypercatabolic state like sepsis, tumor and all that. And in burn injury what happens? You are losing fluid. So decreased water volume content in the body, should dehydration sort of thing is happening. So there will be urea content will be more. Urea will not be filtered properly. And another thing is protein is breaking down. So their hypercatabolic state, for there also urea is producing more. Okay. Hypercatabolic state, intake protein increase to the, if it increase then urea is more, and other than that increase cortisol level and synthetic cortisol analog. Cortisol, what happens? It increase the stress and increase the breakdown of the protein, and it for the neoglucogenesis. So the urea formation will be more. Did you get me? Get it? For the cortisol also, increased stress, increased breakdown of the protein, and protein that will from protein it will form amino acid, and from amino acid it will go for neoglucogenesis. So due to protein breakdown, there also urea will be more. Okay, and other than that, you have a back urea. What happens if you urine flow, if you stop somewhere, so urine will be going to the kidney, like nephrolithiasis, malignancy or prostatism, there also you can get more urea. Urea is not secreted, I mean urea is secreted, it's not coming out from the body, so it's going back into the blood from kidney. So it's like there also you can get more urea. Malignancy, prostatism and nephrolithiasis. In super specialty exam it had come, one of the question is, if there GI bleeding is there, from blood protein also urea can increase. If GI hemorrhage, GI bleeding is there, from that also urea will increase. Any doubt? Prostate, there will be urea will not come out properly, decreased flow. So there will be urea will not be excreted properly. So backflow, due to that urea will be increased in the body. Prostatism means increased of prostate gland. Prostatism. In malignancy, if like it's urethral malignancy or all that, there obstruction due to that, and another point can be hypercatabolism due to broken down of the protein. So that can be also causing increased urea. Another thing, congestive heart failure, their renal perfusion will not be properly done. So there also urea will be increased. Now done. Okay. What we do in modified Berthelot method, that is a end-point reaction. End-point reaction means there is too much affinity for substrate and enzyme. So the reaction will be fast and there will be equilibrium. In that point, so we are incubating for some time and there we are measuring. So we are giving some time for reaction to be finished, then we are measuring. So that is the end-point reaction. Enzyme, substrate, affinity is too much and there will be equilibrium, and there we are measuring the here we are measuring the urea. Okay, and too much, it's more. There is two method, kinetic method and endpoint method, and modified Berthelot is endpoint method. Another thing you have to know, during procedure we can heat it for faster reaction, or you can keep it in the incubate in the normal room temperature, but you have to give more time. So we the reagent and all that we heat it in hot water bath. Now principle, urease, in Berthelot method we use urease. You get the urease from fava bean or arhar dal. Okay, you get that urease. Urea present in another thing where we measure the urea. We measure the urea in blood, serum and plasma. And they had asked what is the difference between serum and plasma. Can anyone tell? Yeah, fibrinogen will not be there. Okay, so you have to tell that if they ask you. Okay, then urea, in presence of urease will form ammonia and carbon dioxide. Okay, and this ammonia will react with that chromogen with the all that buffer and all hypochlorite in presence of that, and it will form a green colored complex. That intensity of the color increase with the presence of the urea in that sample. So if it's more greenish, means it's more urea is present. Okay, so this is the principle, increase urea, increase color, and you have to remember the reaction from urea. With the help of urease, it will form ammonia and carbon dioxide. Ammonia with phenolic chromogen plus hypochlorite, it will form green colored complex. So urea is green. While doing the Berthelot method, you will get the green color. The intensity of green color will tell you the presence, how much concentration of the urea present in the sample, and it will ammonia will react with phenolic chromogen and hypochlorite reagent and will form that green color complex. This is the reaction. Normal level is 14 to 40, 15 we say, but okay. In literature kit they have written 14 to 40 milligram per dl in serum, and in urine they have written 20 gram per liter. That is normal. So 3 sample we can measure: blood, plasma and serum. Plasma, serum and urine. Sorry, plasma, serum and urine. From blood only we will get plasma and serum. So plasma, serum and urine. Okay. So normal level of urea 14 to 40 milligram per dl in blood, and in urine is serum, and in urine is 20 gram per dl. What? Nothing. Okay, fine. There you will see 3 bottles. Okay, one is for buffer, one is for reagent, and another is chromogen. Chromogen will give the color, that is phenolic chromogen, and in the buffer, okay, that is phosphate buffer is there, and the composition, if you want to know, 3 bottles are there. One is written buffer, diluent, and another is reagent, and another is chromogen. 3 bottle will be given. In all that bottle, the composition will be phosphate buffer and nitroprusside along with that hypochlorite and some stabilizer, preservative and detergent. And one standard will be there. For every estimation, they have to give you a standard. So in that aspect, you can measure how much that solute or enzyme, solute is present. So you need a standard. Okay. Now we will measure this thing. You will remember that light path will be 1 centimeter, and if I don't write that, L1, L2, distilled water, sample and standard. 1000 microliter means 1 ml. You have to remember that. Okay. You know 10 to the power minus 6 liter means 10 to the power minus 6 liter means 1 microliter, and 1000 microliter means 1 ml. Okay. Do I have to need to repeat? 10 to the power minus 6 liter means 1 microliter, and 1000 microliter means 1 ml. This one you have to remember. Now first L1 we had the buffer. We will take 1000. This is all in microliter. L2 is enzyme reagent. You are giving urease. So most probably enzyme reagent will be having that urease. We will give 100 microliter. Now distilled water for blank 10 microliter. For sample you will give here 10. For standard you will give here. This is B for blank, S for standard, T is for test. Okay. Now, this thing is working reagent. You are making it buffer and with the enzyme reagent, and then you are adding it to see the absorbance. Okay. So, this thing you will incubate for 5 minutes in hot water bath, that is 37 degrees centigrade, near about 37 degrees centigrade. If you do it in room temperature, near about 25 degrees centigrade, you have to incubate for 10 minutes, but we don't have so much time, so we do heat it. Okay, so 5 minutes we will heat it, and we'll put it in the colorimeter or spectrophotometer, whatever. We'll see the absorbance. We'll check the absorbance. Okay. After we check the absorbance, now they didn't give any color. We just proceeded the reaction, but no color formation. Next, we'll add chromogen. There 200 microliter, and we have to incubate because it has to react with it another 5 minutes. Okay, then we will see the color, and we will find the absorbance. Here you don't have to see the absorbance. Later only if you see the absorbance is fine. What the machine will see, absorbance, how the calculation will be done. Absorbance of test minus absorbance of blank by absorbance of standard minus absorbance of blank. So everywhere from test and standard absorbance of blank we will deduct it. Why we are doing that absorbance of blank? Because that reagent can have the color and it can give us some absorbance. So we want to nullify that. For that we are measuring it. Okay. Absorbance only optical density. And another thing, while doing the test, you have to see what is concentration and what is absorbance. Absorbance and concentration is not the same thing. There is two option will be written, C and absorbance, ABS, I think whatever it will be written. You have to check both. You cannot say the absorbance is the concentration. You have to check both. Absorbance and concentration, you have to check both. This is the absorbance and this is the concentration. The final answer is concentration. Anyhow, you have to tell, that is must. But absorbance, if you say and if you tell, you have to know it. That absorbance is not the concentration. Don't write it, okay? Till here you will incubate. After putting chromogen, you will only incubate for 5 minutes. That's all. Okay. You have to write the result. That is very must. Means in a report it's written like glucose is this much 100 milligram per dl. So urea also how much you got, that you have to write. The urea in the present in the given sample is how much. Okay, you have to write that result. You have to write, otherwise it is incomplete. Means you have written that procedure and everything, but how much is present, that result you have to write in separate line. So the reaction is endpoint reaction, and you will measure in 578 nanometer. That filter is 578. Mostly yellow filter, but if you don't see yellow, it's also okay. Colored is green, and 578 nanometer is the wavelength. Another thing, in the all that while working, you have to be cautious, because in that reagent and mixture buffer, all that sodium azide can be there, that is can cause that skin and mucosa irritation. So if you have any spillage, just wash it with water properly. And you cannot have any ammonium salt or fluoride or heparin. Okay, any ammonia from outside shouldn't be added. So any ammonium salt shouldn't be, contamination shouldn't be there. And chromogen, it contains chlorine, so you have to you have to cap it faster. Like, you shouldn't let the chlorine go out in the air. So chromogen you will handle little bit faster. In the chromogen there is a chlorine, so if it goes out, that reaction can be a bit hampered, but I guess it's written here, but we cannot do very fast also. If in hot water bath 5 minutes, and then outside 5 minutes, and if it's not hot water bath, room temperature, if you have to do everything, that is 10 minutes and 10 minutes. So I think after chromogen also it has to be incubated. After chromogen also you have to incubate. 5 minutes, 5 minutes, otherwise 10 minutes, 10 minutes. Here 5 minutes and 5 minutes incubation time, or 10 minutes or 10 minutes. So if it increasing, it has to be incubated. Here it's not mentioned, but I think it has to be incubated. Otherwise they would say 10 minutes and 5 minutes, but they're telling 10 and 10, so it has to be incubated. It's not mentioned here actually, but it has to be incubated. And the kit can measure up to 250, and if it's too much, it crossing the linearity, means it can't measure, then you have to dilute it and measure it. So how you can write? First you can write the method, then principle, reaction, reagent, procedure, result, clinical interpretation, importance. And normal reference range also you will write. I missed something. In procedure, after that there will be calculation. How you are doing it? Result, surely you have to write how much you are getting, and concentration and absorbance you have to be very specific. Don't write concentration is absorbance or absorbance is concentration, that is separate thing. You just see the LCD digital that display and check it that two different things are there. And it's better you ask them and check, see for yourself the kit literature. So that OCA, the mnemonic, ornithine will be in the inside mitochondria of the liver cell, and citrulline and arginosuccinate and arginine will be in the cytosol of the liver cell, and from arginine it will be urea will be formed. So urea concentration will be increased in high protein diet, increased protein catabolism during tumor or burn, all that, sepsis, reabsorption of blood protein after GI bleeding, treatment with cortisol because there is stress hormone, stress during stress, neoglucogenesis, protein breakdown will be there. Treatment with cortisol or its synthetic analogs, and dehydration. Anywhere is dehydration, so urea content is in concentration is becoming more. So any vomiting, diarrhea, it will be more in urea can be more. And other than that, decreased perfusion of kidney, like in heart failure. And other than that, it is malignancy, nephrolithiasis and prostatism, that is post-renal, obstructive post-renal condition. So urea increment, you can say pre-renal, renal and post-renal causes. Post-renal causes are the obstructive, malignancy, prostatism and nephrolithiasis. Okay, and pre-renal dehydration, increase of protein intake, catabolism, vomiting, diarrhea, cortisol over. And in renal cause, you can say acute glomerulonephritis, because their glomerulus will be damaged and waste product will not be filtered properly. So that will be increased urea. So pre-renal, renal and post-renal causes of increased urea. And decreased urea is seen in pregnancy, children and female. And also liver failure. Liver cannot produce. So one is site is liver, another is kidney. So any problem in this post side cannot perfuse or cannot excrete and cannot form. You will read about the ammonia toxicity in the brain, how it's removing and how it is removed from the brain and all that. You will read it in practical class or next class. I will explain it. Okay. You know by yourself, you can come to us or we will tell you. And this is endpoint method. Urea can be also measured by kinetic method also, that is different method, but this is the endpoint method. In that we are measuring urea. There is other kinetic method also, we can measure urea, but they have told that kinetic method we will do later, so we will elaborate that later. And the light path will be 1 centimeter. That colorimeter, you are putting that cuvette or whatever that tube, that light path is 1 centimeter. Okay. 578 nanometer is the wavelength. In that we are measuring the concentration of urea.